Okay, I think we'll go ahead and get started. So good evening and welcome to this panel discussion on biology, mathematical modeling and spread, or the mathematical modeling of spread and data related to COVID-19 with a question and answer session at the end with some of our biology and mathematics and data science faculty. So a couple of housekeeping items first before we get started. I'll just ask everyone to keep their microphones muted throughout. We'll have a chance for the question and answer session at the end after all of the speakers have gone through their presentations, and I'll moderate that through the chat function. So you can enter your questions that way. And I'll be moderating the discussion tonight to introduce the speakers and then kind of field those questions through the chat. So I'm Jennifer Glowinka. I am the Associate Vice President for Academic Affairs at Carroll. And as we get started tonight, I guess the other thing I'll mention is that we are really focusing on the science and mathematics of COVID-19. We're not going to be entertaining any questions on politics or matters of public policy, and so we'll keep all of our questions and discussion related to the science and mathematics of this virus. So with that, we'll introduce all of our speakers tonight. So first, I'm going to spotlight Dr. McHugh. Our first speaker tonight will be Dr. Theresa McHugh. She's an assistant professor of microbiology at Carroll. Her research is on the ecology of soil microorganisms, and tonight she'll speak about the biological aspects of COVID-19. Our second speaker is Dr. Kelly Klein. He's a professor of mathematics at Carroll, and he'll be talking about how we create mathematical models to explore possible ways that diseases can spread through a population. Our third speaker is Dr. Grant Hokit. He's a senior research associate and ecologist who studies the spread of zoonotic disease, particularly vector-borne diseases such as West Nile virus. And tonight he will talk about how virus breakouts occur and how they come to an end. And then our last speaker is Dr. Drs Sullivan, and he's an associate professor of math and data science at Carroll. His mathematical interests are applied math, numerical analysis, and machine learning. Tonight, his part of the presentation will focus on examining some of the data related to the COVID-19 outbreak, looking at how to spot trends in some of the data, and to give a broad overview about how to interpret some of the statistical models that people are using. Okay, so with that, I will turn it over to Dr. McHugh to get us started about talking about the biology of the virus.
All right, thank you, Jen. And good evening, everyone. I'm going to start generally talking about viruses, and then I will transition to some specifics. And so viruses are inert particles that are completely dependent on the host cells that they infect to facilitate their reproduction. As such, we consider viruses to be obligate intracellular parasites. Viral size is variable, but is generally measured on the nanometer scale. So we're talking about this leftmost side of the ruler here, where 1 nanometer is equal to a billionth of a meter. 100 years ago, scientists could not imagine submicroscopic particles, and so they referred to these infectious agents as a contagious fluid. By the 1930s, scientists had started using the term virus, which is the Latin word for poison. All viruses have some common features. One is that they contain a single type of nucleic acid. This can either be DNA or RNA. And then that nucleic acid is surrounded by a protein coat or capsid. Some additional features are possible. For example, most animal viruses contain an exterior membrane that is made of lipids and other biomolecules. This is called an envelope, and it plays an important role in attachment to host cells. Coronaviruses are a large family of viruses that usually cause mild illnesses like the common cold. Coronaviruses get their name because when viewed under electron microscopy, each virus particle is surrounded by a corona or crown. Coronaviruses are really common in humans. They're also found in a variety of animal species, including pigs, camels, cats, and bats. Very rarely, animal coronaviruses can infect people and then be spread between people. Examples of viruses that have done this include SARS coronavirus, which is severe acute respiratory syndrome coronavirus. Also, MERS coronavirus, which is Middle Eastern respiratory syndrome coronavirus, as well as the virus that we're currently dealing with, which is called SARS coronavirus 2. And all of these viruses had their origins in bats. So SARS coronavirus circulated in 2003. It caused 8,098 infections and 774 deaths. MERS coronavirus has infected 2,494 individuals since 2012, and it has caused 858 deaths. As of today, SARS coronavirus 2 has infected over 2,176,164 individuals and caused over 145,304 deaths. So SARS coronavirus 2 first emerged in the later part of last year in Wuhan, China. By February, the World Health Organization had issued an official name for the disease that is caused by this virus. The name, Coronavirus Disease 2019, abbreviated COVID-19, makes no reference to people or animals or places so as to avoid stigma. SARS coronavirus 2 is believed to spread primarily from person to person via infectious respiratory droplets that are emitted from an infected individual when that person either coughs or sneezes or simply exhales. Likely you already know a lot about this aspect. And so it's possible for infectious particles to land on the mouths or noses of people who are nearby, or even to be possibly inhaled directly into the lungs. This kind of spread is most common when people are close to one another, like within 6 feet of one another. It's also possible for transmission of infectious material to be transmitted directly through contact, such as handshaking, or indirectly through touching surfaces that have been contaminated or objects that have been contaminated with the infectious material. The common symptoms of COVID-19 include fever, dry cough, fatigue, muscle ache, as well as loss of appetite and possibly loss of smell. In some individuals, more severe symptoms develop, and these can include high fever, severe cough, and shortness of breath, which often indicates pneumonia. The virus targets the lungs. However, lack of oxygen and very widespread inflammation can negatively impact the kidneys, the heart, the brain, and other organs. It's possible for infected individuals to show no symptoms at all, or for very mild symptoms to be present for some time, followed rapidly by a worsening of the condition. Recently published research indicates that the time from exposure to onset of symptoms on average is 5 to 6 days. However, some studies have found that symptoms can develop as soon as 3 days post-exposure to as long as 13 days after exposure. And collectively, all of this work continues to support the CDC's recommendation of self-quarantine and monitoring of symptoms for 14 days post-exposure. I've noticed a lot of confusion regarding testing. So COVID-19 is a respiratory disease, and testing therefore has to start by collecting a sample from the respiratory tract of a patient. This is usually done through a nose swab or a throat swab. Some people think that having a specimen collected means that they've been tested. But actually, the test is much more complicated than that, and it involves a very complex set of procedures that are carried out in a clinical microbiology laboratory. The primary test that is involved is called real-time reverse transcription polymerase chain reaction, or RT-PCR. SARS coronavirus 2 is an RNA virus, and so the procedure begins by extracting RNA from the sample that has been collected. That RNA then gets turned into DNA using an enzyme called reverse transcriptase, and that is the RT part of this procedure. Then the DNA can be copied or amplified using specific DNA sequences called primers that are unique to this particular virus. And that's the PCR part of the test. This test is very accurate. However, it's also time consuming. It often takes a day or two for the results to be visualized and then shared with the physician. This test is only effective when done on a person who is currently infected with the virus. And so if you've had COVID-19, but you're no longer showing symptoms, then this test that is designed to detect viral RNA is going to tell us nothing. In that situation, we need a different kind of test that is called a serological test. And this test will look specifically for the presence of antibodies that the human body has created in response to the presence of this viral invader. And so antibodies have the effect of neutralizing the virus. They can also serve as a protective role that can prevent the development of disease again later on if the virus is encountered. And so a serological test is a test for the body's response to the virus, rather than a test for the virus itself. The catch here is that it takes time for antibodies to form. And so this type of test is not helpful for us early on. However, as the outbreak continues, we're going to want to do serological testing by drawing blood on as many individuals as we can, so that we can identify those people who were previously infected and who are very likely to be immune. And those are the people who can safely return to work. The tremendous impact that the COVID-19 pandemic has had on humans and on the economy have been a very strong driving force behind vaccine research and development. As of last week, there were 115 candidate vaccines that were available. And among the most promising of these is the product of a collaboration between the National Institute of Health and a private company called Moderna. So this particular vaccine entered into the first stage of clinical trials on March 16th. And remarkably, this was just 42 days after the viral genome sequence was made public. In general, vaccines are a simulated infection. And so by inserting non-infectious virus material into a human recipient, we can trigger an immune response that involves the production of antibodies as well as memory cells that will prevent the development of infection later on if the person comes into contact with the virus. A very strong target for antibody production against this virus is the spike protein that is found on the surface of the virus. And in the case of the vaccine that I mentioned that's now in the clinical trial, this is an mRNA vaccine. And so this vaccine inserts into human cells via an injection the instructions for making that spike protein. And so the human cells that receive these instructions will make the spike protein as they would if they were actually infected with the virus. Immune cells can then interact with that foreign material and develop a long-lasting immune response, a memory response, such that disease will not develop later on in time when the virus is encountered. And this is a really cool type of vaccine, and it's a new type of vaccine. With the advanced stages of research and development that are at play, it seems possible that we might have a vaccine available for emergency use as early as the spring of next year. Thank you all.
Great, and with that, thank you, Dr. McHugh. We'll transition to Dr. Kelly Cline, who will be talking about how we create mathematical models to explore the possible ways that disease can spread throughout a population.
Excellent. All right, so now we know a little bit about what the virus is. How do we predict how this is going to spread? How do we predict, what to expect in terms of numbers of people sick? How do we do that with any sort of disease? Well, first of all, let me emphasize that this is a really difficult mathematical epidemiological problem, fiendishly difficult. So the fact that we can do anything at all is rather miraculous. So the first thing we do is we say, well, trying to figure out exactly which person is going to get sick is going to depend on who sneezes on who and who's in what place at what time. Nobody can predict that. That's ridiculous. That's something far beyond anything what we can do. So our strategy is let's just talk about large numbers of people, thousands, millions of people, and hopefully the factors in play will be easier on average. So our strategy is to start simple. A scientific principle of Occam's razor. Usually the other things being equal, the simplest solution is usually the right one, or perhaps the most useful. And what we're going to do is we're going to create mathematical models. Now, a mathematical model is not trying to predict reality. Reality is big and messy and complicated, and there's no way in the world we can do this with any great precision or anything at all. But it may allow us to kind of see what's possible, to explore how our ideas of things and various trends can play out if they continue. So that's what we're going to do. All right, so let's play mathematical modeling with this. Here's about the simplest possible thing we could do. Just suppose as an exploratory scenario, suppose we start out with 10 sick people and we assume that on average, each person spreads the disease to 2 other people after an incubation period of 1 week. So as a result, for every week that goes by, the number of infected people will triple. this will lead us to what we find as an exponential growth scenario. Since every time we go from 1 week to the next, we have triple the number of sick people we had before, then this is going to go up by a factor of 3 every time. If I want a function to describe this, that would be 10 times 3 to the power of how many weeks it's gone by. And we find that sometimes, at least in the initial stages of disease or an infection spreading through a population, sometimes this actually isn't a completely horrible model. But we can also see that it goes pretty wrong pretty fast. Okay, so after 1 week, we've got 30 people, then 90 people, then 270 people. By week 10, we would predict that 1 million people would be infected. By week 20, we'd predict that 34 billion people would be infected. And of course, that makes no sense at all. I mean, that's way more than the number of people in the world. So exponential growth may model an early phase of the growth of things, but that's certainly not going to work very well for any period of time. Also, this model never accounts for anybody getting better, so we need to think a little bit harder than that. Again, we want to stay with a fairly simple model, try to just make very basic assumptions, and then see how they play out over time. So the next thing that we do is we go to what is called an S.I.R model, where we group people into 3 different compartments, 3 different places where we put them as susceptible to get sick, infected, these are the people who are actually sick and potentially spreading it around, and then recovered, and it would be lovely if the recovered people would then be immune and then they couldn't get sick again. So if there's kind of the simplest possible way to do it, if we're going to include people getting well, and then maybe even limit things so we can't get 34 billion people sick. This model was first published by a couple of Scottish researchers in 1927, and it has been used a lot. And again, the goal here isn't to predict exactly what happens. That's too hard. The world is big and messy and complicated, but perhaps we can gain some insight into what sorts of things are possible. So we take each person and we put them in one of these categories, and we can do this in terms of raw numbers of people or as percents or decimals. And then the real question is, well, how will these numbers change? How do we decide at what rate people are going to move from susceptible to infected, and then from infected to getting better, to recovered? So that kind of is the heart and soul of the model. So this is what those Scotsmen did 100 years ago, as they said, okay, we're going to need a constant in there, so we'll call it beta here, which is that's going to vary depending on lots of factors, the virulence of the virus, and then the how people are interacting with each other. We'll talk about that more in a minute. And then, well, the rate at which people are getting infected should be proportional to the number of people who are sick. The more people who are sick, the more people are out there sneezing on other people. And so that's going to, the bigger I is, the faster I will grow. But then we also need to limit growth. We need to say that, okay, we can't grow to more people than we've got. So we can also multiply by a factor of, here I'm going to do it with S, the number of susceptible people, over N, the total population. And so that's kind of a fraction. So at the very beginning, basically everybody's susceptible. S over N is about 1. So we just have, this leads to an exponential growth process. But when most people have been sick and there aren't that many susceptible people left, then this fraction S over N will be small, and that will prevent our model from predicting 34 billion sick people. So again, the idea is, let's create a simple model, not one that includes all the possible effects, but something basic to just give us a sense of what might happen. So, and then the other rate we have to include is the rate at which infected people recover and get better. And so we're going to assume again that that's going to be proportional to the number of infected people. The more infected people are out there, the more are going to be getting better. And then we'll put a constant on that, gamma, which will describe how quickly infected people recover. This is going to depend on lots of things, not only on the properties of the virus itself and the human immune system, but also depending on things like how well people take care of themselves, the quality of medical care people receive. If there's better, they'll be getting better more quickly, hopefully. And so this is, again, the idea is to create a very simple model. So we put all our people in one of these 3 categories, and now we have described some very simple assumptions we can make about how quickly they move from one category to another one. And so we put these together, and here are what we call 3 differential equations. 3 equations to describe the rate of change of these things. So here, notice I've got S and I and R, and then I got a little prime, a little apostrophe sort of thing next to them. Those symbols indicate I'm talking about the rate of change of this. How many people per day or per week or whatever I'm working with are going to move from one category to the other. So for the susceptible category, you'll notice this is just a purely negative number, minus beta and the I and the S over N. That's going to be negative. So what we're saying is that once somebody's susceptible and then they become infected, then they don't go back anywhere. So that number is only going to decrease over time. And then for the infected category, you both have that term now positive coming in, bringing people into that category from the susceptible category, as well as we're subtracting people out of the infected and spreading disease around category into the recovered category. And then we see it's a negative there. And then that same term, the gamma I, is positive on the R prime. And so this together characterizes a simple, a basic S.I.R model. And so what I can do is now, if I know if I choose some initial values for, okay, here's the S, the total number of susceptible people in my population, here's the starting number of infected people, starting number of recovered people if we have any to begin with. And if I then pick numbers for beta and gamma, I can use math. I can now make some predictions about what will happen. I can extrapolate what will happen and how these will change over time. Again, it's a model. It's a toy. The idea is to explore what's possible and what would be out there if we see what happens. So here we've got, in the lower left, we got a bit of a graph where we can see this blue curve is the number of susceptible people. And we can see that in a typical S.I.R model where we pick a value for this and that, notice we started the infected people, that rises up over time and then gradually tails off again as people get well. And then we end up with more people over in the recovered sorts of thing. I guess what I find most amazing is, this is a really fairly simple model, and yet it has been used very effectively. There have been cases where epidemics of influenza or dengue fever, SARS in 2003, people have used models like this in order to explore and to see what's going on there and to see what has happened. But the important thing to remember is when we're doing this, we've made some really big, really simplifying assumptions. These numbers, these beta, the getting sick rate, and gamma, the getting better rate, these depend on a lot more than just, the biology of the virus itself, but also on human behavior. When we start social distancing, well, that means people are going to be getting sick at a lower rate. So that's going to change things. And if people learn and change their behavior or change it back, this is going to be a really complex sort of thing. So mathematically, all we can really do is make some estimates of what are reasonable numbers for these and then extrapolate them forward to kind of get a range of possibilities, what can happen. And from the beginning, it's very, very difficult to know what these numbers would be. So we start out with some data. We see how quickly it spreads in China or where we have places where we have data, and then we can make some rough estimates of this. In general, this is easier to do long after the epidemic or pandemic has taken place and then try to fit the model to the data. It's a lot harder to do under the gun when people are really interested in what will happen. Seeing where exponential growth turns into a leveling off, that's a difficult sort of thing. If an S.I.R model runs with constant beta and gamma and we let it go all the way through, well, then we end up over here with basically everybody recovered. That's the herd immunity sort of thing that some people talk about. But it's hard to say exactly how that will happen. So, and then once we get started with this, there are lots of other things we can do. We can add another category E of exposed people who aren't yet infecting other people. That's something that's been done very effectively. Sometimes when people are recovered, after a while the immunity wears off and we have to put them back into the susceptible category again. These are the sorts of things that epidemiologists and mathematicians do to try to understand and make sense out of it. We can then add, to take it to the next level, now instead of treating this as one pool of people, now we can add geographical information and say, okay, we're going to run an S.I.R model for all the different communities throughout a nation and then make some assumptions about how they interact with each other. There have been some published models that worked on that, tried to make models of how this would spread throughout an entire country. But again, still based on these S.I.R ideas. And we find that if we can change things so that beta falls low enough, then things will die out. So that's why everybody's all talking about social distancing these days. But anytime we change that, these numbers will change. They're not constants. They're constants when we put them in our models to explore what happens. But if we do a lockdown, beta goes down, we're interacting with each other less, the disease might start to die off, but then ending that will increase this again. And that's why this is a very, very difficult thing to do. It's based on so many different factors. It's really hard to predict how this is going to happen and how this will proceed with people constantly altering their behaviors and how all this will work out. So trying to predict the spread of this and how this will develop ultimately is a very difficult thing. And I have great sympathy for the people who are doing their very best, putting some models out there in the public for everybody to look at, everybody to make decisions based on and critique in all their different sorts of ways. It's not easy science to do, but it's really vital these days. And the fact that we have people brave enough to put their models out there in public view, I think is amazingly useful to all of us, even if the models themselves are very difficult to interpret and can only at their very best give an approximation of what sorts of things might happen in the future. All right, well, there's an introduction to that. I think we move on to our next speaker.
Wonderful, thank you, Dr. Cline. That we will switch over to Dr. Grant Hokit, and he's going to talk about how these virus outbreaks occur and how they end.
Okay, so earlier in the presentation, Dr. McHugh mentioned how sometimes, rarely, a virus that starts off in an animal species will jump host to us humans. That's what we call a spillover event. Excuse me. So one of the conditions that at least promotes a pandemic, it's not a requirement, but it can certainly promote a pandemic, are these spillover events. And this is because the spillover pathogens are novel pathogens that are being seen by the new reservoir host, humans, for the very first time. And humans in this case have not developed any form of acquired immunity against this particular pathogen. Sometimes these spillover events can occur with a couple of jumps. A virus starts in one reservoir host animal species, jumps into another animal host, and then eventually arrives at humans. They're relatively rare events for any particular virus, and even rare when you compound that with the virus then has to be transmissible between humans. But if that event happens, it's then just a matter of time before these events can be spread globally. The second condition that makes pandemics even more likely are increasing the transmission opportunities, which the map on the left shows you quite vividly how global transport by airlines has enabled transmission of pathogens once they are acquired by human hosts to become quite effective. The graph on the right indicates numbers in millions. So for 2019, for example, 4.5 billion humans traveled by air. And if I just draw your attention to Europe, you can see it being a major hub for all of this world travel. It's not really a surprise that COVID-19 really exploded in the European countries. A third condition is the prevalence of zoonotic diseases that we know are out there in the environment. Even though, as was stated earlier, it's a rare event for a virus to jump from an animal reservoir to humans, and then even rarer for it to be transmissible by humans. The sheer numbers of viruses that we now know exist out on the landscape is quite phenomenal. If you look at this particular study that was mapping the probability of outbreak events, you can see that most of those events are concentrated in tropical regions and subtropical regions, such as the Hubei province in China. This isn't too surprising to us zoologists, because if you look at the spread of biodiversity across the planet, we know of very precisely tens of thousands of virus hosting mammals alone that exist in the tropics. This includes, of course, bats, rodents, and other species. If some of these bat species, we've quite carefully analyzed the viruses that we host. And if we extrapolate out that each of these bat hosts can host somewhere on the order of thousands of viruses that have the potential to be transmissible to humans, we extrapolate that out across the number of bat species and across the tropics. We're talking about hundreds of thousands of viruses that are waiting to spill over into humans. This isn't really a surprise to us scientists. COVID-19 was not a surprise. For decades, we've been observing that as humans encroach more and more upon these tropical regions, the episodes of zoonotic outbreaks have increased. If we go back to the 70s, you all will recall names like Lyme disease. You'll recall diseases like HIV, AIDS, avian flu, Hendra virus, and West Nile virus, the virus that I happen to focus on. That enticed some epidemiologists in 2001 to write a paper warning society that these outbreaks seem to be on the increase, the zoonotic spillover events are on the increase, and that we should take warning and prepare. Subsequent to that 2001 paper, we've experienced SARS-1, MERS, and several other viruses, the H1N1 outbreak, and now we're dealing with SARS-CoV-2, the virus that causes COVID-19. So if those are the conditions that are conducive to a pandemic, how do pandemics end? Going to Dr. Cline's presentation, we can conceptualize, it's relatively easy conceptually to end diseases. If we look at his bins of susceptible, infected, and recovered individuals, all we really need to do is break the chain of transmission between infected individuals and susceptible individuals, and we can end the pandemic. Putting that into practice is quite difficult, however. How to end the transmission between infected and susceptible individuals? In general, there are three main strategies. The first one is that we could try to survey and test and detect infected individuals, isolate them, and therefore contain the infection in a small geographic region before the virus is able to be transmitted to larger susceptible populations. Obviously, we've failed at that in the case of COVID-19, although we're trying to reset the process with our stay-at-home orders. If we drive the case rates down low enough, we can then re-implement testing and try to isolate and contain spot fires and outbreaks of the infection that will be surely developing in the next coming months. The other strategy that we could employ is, as Dr. McHugh pointed out, the development of a vaccine. We can develop vaccines and immunize susceptible individuals, and this will provide the herd immunity that we are referring to, in which the virus will have a very difficult time being transmitted from host to host. Of course, the problem with that strategy is that developing vaccines take time, and we are months, if not years, away from having a viable vaccine. The third option is not the most palatable option, and that is that we can increase herd immunity by having susceptible individuals contract the disease, go through the infected stage until they either survive and become recovered. Of course, the obvious problem there is not all individuals will recover. In some populations, like with COVID-19, the elderly and the immune compromised are going to be particularly highly impacted, resulting in tens of thousands of deaths. So when will COVID-19 end? If we apply those general conditions, COVID-19 will end when we can test and isolate infected individuals, and or when we develop an effective vaccine, and or when the majority of us contract the disease and hopefully recover from that disease. And I think I'll stop there and turn it over to Dr. Sullivan.
All right, thank you, Grant. All right, so let me share my screen out. Okay, so what I want to do is I want to take a look at the data that's come out of COVID-19. And really there is an absolute wealth of data that's associated with this outbreak, because Every country is interested in gathering the data. Every country is gathering data. The World Health Organization, the CDC, and all of the other similar sorts of entities are all interested in the data. And it comes down to, well, we want to be able to build good models so we can make good public policy decisions, so we can make good decisions about how we handle things. And going back to Dr. Cline's and Dr. Hokit's presentations, we also want to have good data so we can support the mathematical models that can predict further into the future. So what I'm going to do is show you a couple of things that I think are kind of reliable sources for our data. One of them is this Johns Hopkins data dashboard. And so I'm going to jump into that dashboard here. Give it a moment to load. We'll give it a moment to load. So the type of plot that's going to show up here in the middle is what's called a dot plot, and it should show. We'll try one more time. If it doesn't show, we'll move on. This particular dashboard, anybody can Google this, just search for Johns Hopkins COVID-19 data. And it doesn't want to show me the picture, which of course it did every time I practiced this, so whatever. You'll see where the hotspots are around the world. And you'll see where, well, hypothetically, you would see where the hotspots are around the world. And those are all actually listed by country, but you'll notice that the dots are actually oftentimes just one per country. Like there's one here for Brazil, or there's one here in India. Well, they're just putting a dot on the individual country that's gathering that data. That doesn't mean that's how the geographical spread is. Now, when there's more counties and states in the country gathering data, like the US and like in Europe, you'll actually find a little bit more granularity. Now, most of this data is gathered from the World Health Organization, from the CDC, and then partners of those institutions all around the world. And so I've put a couple of those here, and I noticed that Dr. Glowenka actually put the link to the map over in the chat. And that's a good reminder too. If you have questions, you're welcome to jump into the chat and ask those questions too. So thinking of Dr. Cline's presentation and thinking of Dr. Hokit's presentation, it stands to reason that we should look at what's actually happening over time. And you hear, or at least we heard for a long time, that exponential growth was how this was being described. Well, exponential growth is really what Dr. Cline explained, where we start off with a small number of cases, and every week or so, we multiply by another factor of 2, let's say. And so after a week, we have 2 cases, then we have 4, then we have 8, then we have 16. And initially, exponential growth doesn't actually seem to be growing very fast. But actually, over just a handful of weeks, we can more than exceed the population of the entire world if exponential growth were to just continue forever. So I've got some plots here that hopefully can let us see what exponential growth looks like and what it doesn't look like. So this is the US per 100,000 people, so per capita. And initially, in this region of the plot, you can see that it's actually starting to take off like a wildfire. But as time goes on, you see that it actually doesn't seem to be growing at kind of the same rate over here. It almost looks like a linear growth. So if I put this on a logarithmic scale, so that means every one of these white ticks is actually 10 times higher than the other. So this is 1, there's 10, there's 100. And you'll notice that on a logarithmic scale, the US is actually starting to bend over. So over the last week or so, we've stopped exhibiting exponential growth, and we're starting to exhibit what's called logistic growth. So, okay, what is logistic growth? Exponential growth can't go forever. We simply can't have it go forever. What happens is that you reach some sort of carrying capacity in the population. So I've got the plot here for China. China has already seen their main peak. Now that's not to say they won't have another, but you can see that over time their cases per capita actually rolled over and have reached some sort of carrying capacity. But unfortunately, what you see now is that it's starting to actually grow again, because China is starting to loosen up some of their restrictions on keeping people at home, and it's going to start growing again. So the goal of social distancing, for example, is to really get this exponential growth curve that we looked at back here to start to roll over so that we don't have such a fast, rapid growth. Now, one thing as a mathematical modeler that you learn very early is that plotting against time can be really deceiving. You can fool yourself into thinking that there are correlations that aren't actually there. So I'm going to speak out of both sides of my mouth. I just showed you 2 plots over time. And now I'm going to say that might not have been the best idea because you can learn more with this tool that us mathematical modelers use all the time. Here I'm plotting the rate of change of the cases compared to the number of confirmed cases. So what I'm looking for here is how the rate of change compares to how many people are currently confirmed. And early on, you can see most of the countries that I have plotted here are actually exhibiting some sort of linear growth, which means the rate of change is directly proportional to how many people are sick. Well, of course that's the case. That was before we were putting in really good social restrictions and keeping people kind of away from each other. Now, one thing to keep in mind is that time is implicit here. So time is not from left to right. So this little blip down here, this pink guy, that's South Korea. They had that linear growth and then dropped off when everybody started to just stay at home. You can see Spain has dropped off. Italy had that linear growth and then dropped off. Their rate of change relative to the number of people who are confirmed is actually starting to turn the corner. Now the US is this orange one. I gathered this data 2 days ago. The US is the orange one coming down the middle. We have not started to see the point where we're going to turn that corner yet. We're just not there yet. So the last thing I want to leave you with is that all models are wrong. It's a classic saying by statisticians, but it goes on to say all models are wrong, but some models are useful. And in this particular case, these models that are wrong, you need to be very wary. I'm trying to give you like the public service announcement here. You need to be very wary of listening to news reports and having them give you one number and say, oh, that's the model, and not think that every statistical model has to have some sort of margin of error associated with it. So the picture I have here on the right is from healthdata.org. They're actually plotting out the resources that we're going to need to take care of COVID patients. But you see this giant purple band around their model. Now, first of all, the model itself looks a lot like Dr. Kline's infected group in his S.I.R model. That's exactly what we're looking at, but we're trying to predict that forward. And this giant purple band says, well, the prediction is anywhere between way down here and way up here. There's a massive amount of uncertainty in the models. So I'm going to try this again and see if, I don't want questions yet. I'm going to see if we can actually get this one to work. So they're updating this every couple of days. So here's what it looks like for the US. And if we go to other countries, so for example, if we go to Sweden, this is what we see. Now, Sweden is doing something rather interesting in that they have not been forcing as much social distancing and social isolation as the rest of the world has. And they're going to see, at least projected by this model, a fairly large peak. And the horizontal purple line is the hospital capacity. They're going to, we're predicting that they're going to see a fairly large surge that's going to overwhelm their hospitals. If we go more granular, we can be a little self-centered here and say, let's look at Montana. Okay, the picture is a little bit nicer here, right? We don't have that much of a big blip there, and it looks like our peak is, oh, just a handful of days from now. But if I scroll down, we also have models for total deaths. And this does not paint that wonderful of a picture. Right now we're around 9 or 10 deaths. This was gathered, April 12th, so 4 days ago. But look at the error bars. This is the thing I want you to take away from this, is that every statistical model for these sorts of things is going to have a large amount of error because we don't know how humans are going to continue to behave. That's what Dr. Cline was talking about, is that these models are awfully devilishly hard to pin down when you're literally talking about each individual human's behavior. So I hope that over the course of this, we've given you a couple of really good things to think about and some really good food for thought. And I think, Jen, at this point, we're going to open it up for questions.
Great. Thank you all very much. And if we've got questions, I would love for you to type them into the chat. Okay, so we've got a question here. On February 22nd at the Distinguished Scholar Lecture at Carroll College, Dr. Hokit gave a public presentation that in part discussed the novel coronavirus before it had spread substantially in the United States. What has substantially changed between then and now, do you think, in terms of your presentation? Is the mortality rate still about 1%, you think? Has anything else changed?
Just for the record, I didn't type in that question for myself. Thank you, Jeremy. Nothing much has changed in terms of our estimates of mortality. Keep in mind that mortality rates are kind of a fluid number, because as we get better and better at treating the pathogen, we inherently drive the mortality rates down. So if you're looking at a raw mortality rate, we're still estimating that it's about 2.5% in the overall population, much higher in the elderly. However, as we've learned more and more about this virus, we're becoming better and better at treating it. And we've driven that mortality rate down in certain regions. And so the only thing that's really changed, I guess, is that we're becoming a little bit better at treating the virus, but the innate mortality rate we estimate is in that same range.
Great, thank you. So the next question is, what is the availability of these tests that detect both virus and antibodies, complex as they are? Are there only a few places, or are they fairly commonplace? Seems like we have a lot of tests, and then the next day there are not enough tests. Can any of you help answer that?
Yeah, I can try to answer that one. And so the availability of tests really has a lot to do with the particular location, and it's variable from location to location. And so in Montana, presently, my understanding is that we have enough tests available to test those individuals who fall into a certain category. So those who are at high risk because they have underlying health conditions, or those who are believed to be highly likely to have encountered an infectious individual, or those who are on the front lines and at risk for receiving a high viral load. So not everybody who wants a test can get a test. And that has to do with the real-time RT-PCR type of test that I described earlier to identify the presence of viral RNA. In terms of the serological tests that look for antibody presence, those are more broad-based surveillance type of tests that I don't think are readily available for us now, but will likely be implemented later on as we try to get a feel for who has been infected and recovered.
So that actually leads into the next question that we have is, what information do we have about whether or not one becomes immune after recovering from the virus? Because that's what what an antibody test would really tell us, is whether they've been exposed, right? And so do we know about the immunity after exposure?
I don't have a good answer for that as to whether the immunity conferred would be a months-long type of immunity or a multi-year type of immunity. We really don't know, as this is a novel virus that we've encountered, and we are immunologically naive in terms of our ability to deal with it. And so we'll only have an answer to that question as time progresses and we can make those measurements.
Thank you. Okay, so we've received a couple of other questions. So the next one is, how does the very first animal get the virus?
Yeah, that's a good question. That's kind of a chicken or an egg question. There are, for every host animal, we each innately carry many microorganisms on us that have been with that particular species ever since the species emerged. So this is kind of a grotesque statistic that I like to share with my students. Some microbiologists worked out a few years ago an estimate that your average human, if you weigh yourself, you can subtract 13 pounds that are the result of the microbes that live in and on you. And most of those microbes are relatively inert and don't affect us. And in fact, some of them are quite beneficial. So each species carries, we're packaged full of microbes already as we emerge as a new species. So where the viruses first come from, that we could take that argument all the way back to the beginning of life itself. What we do know is that there are millions and millions and millions of viruses in our environment. Fortunately, most of them are not in any way pathogenic.
Thank you. Okay, so then our next question is, what type of data, if any, do we have on reinfection or a second surge of infections? And in particular, the person asking the question is referencing South Korea, who has announced several second infections. Which I think then actually goes to another kind of follow-up question that someone else has, which is, you know, if we have these, if I understand the question right, if we have these second infections, and if we don't have kind of longer term immunity after having been infected, is universal testing then really a valid option for ending the pandemic?
Well, so the data about second infections is very, very new. So whether or not those are actual second infections or whether it's something else, I think that's still being sorted out. So at the moment, the answer is we don't know.
Okay. All right, then I've received another question. So can any of you, especially I guess specifically Dr. Cline, Dr. Sullivan, can you comment on how any of the models incorporate a supply chain variable? For example, a shortage of ventilators is a lagging limiting factor, where a break in the supply of PPE is a more near-term variable. Is there any way to factor that into those models?
Well, so piggybacking on Dr. Cline's models, the S.I.R models are kind of the simplest of an entire family of models that allow you to build as many other compartments as you like. So I think at the end of Dr. Cline's presentation, he had susceptible, exposed, infected, recovered, and possibly quarantined. But you can also build compartments into those models that allow a lag time for somebody to not show symptoms. You can change infection rates for certain populations. Those models, since the 1927, Dr. Cline, that they were first published, have been shown to be very, very flexible and can actually capture a lot of that information. The rub is exactly what Dr. Cline said. This is really hard to do in the heat of the moment. So Kelly, you want to piggyback on that?
Well, yeah, absolutely. Right. It's one thing to look at some data from the spread of disease from a couple of years ago, and then try to fit your model to it and tune your parameters and figure out what numbers represent it. Right now, when we really want to know what things are going to look like in a month or two, that's a much more difficult problem. I'm just really glad we have people are willing to step up and try their best at that. That's right. And we've tried to introduce just the most basic ideas behind this. These are, right, the beginning of a whole, there's a wide variety of models that are descendants of these S.I.R models that allow us to account for all these different factors in various ways.
Great. Okay, and then the last question that I have here is, do any of you know what the false positive and false negative rates are of our tests and what that means for the estimates of the true prevalence of the virus in the population?
Gosh, I don't have an actual number to report here, but in terms of the RT-PCR diagnostic panel, that is a very accurate test that is performed by well-trained individuals in the clinical laboratory setting, where they're including positive controls and negative controls in every run, as well as inserting well-curated blinded samples to ensure that the outcomes of each of these tests are as they should be. The second part of that, I would say it's complicated by the fact that not everybody who would like to be tested is getting tested, and also that somewhere around 40% of infected individuals don't show symptoms. And so determining the true number of people who either are presently infected or were infected is very difficult to achieve.
Thank you all. Do we have any other questions for our speakers tonight? Okay, I'm not seeing anything, and we are at 8:04. Oh, so I would ask, everyone to give you a round of applause, but that's a little difficult in these circumstances. So do that. But thank you all for joining us tonight. And anyone has any follow-up questions, I'm sure that our speakers would be happy to answer those via email. So thank you all, and have a good night.
Thank you.